Hello, everyone. Every day, zero point 0.5 million women get pregnant. They may be divided by country, nationality, race, religion, many different things. But one thing is common about all of them. All of them suffer from our inability to predict which medicine is safe for them or not. As a result, for an ailment, even if the medicine is there, the pregnant women are not able to take them for their condition. This situation has developed because of a very peculiar reason. That is, the during the drug development, the safety of a drug is determined through the clinical trial in which patient as well as healthy individuals participate. However, pregnant women are excluded from that drug safety testing that is during the clinical trial because of the obvious ethical reason. But as a result, we do not have enough evidence to know which drug is safe or not during pregnancy. As a result, out of all the medicines that we have, 8 drugs are known to be safe, 12 drugs are known to be unsafe, and rest are unknown. Now you think of a situation that a woman having a chronic condition, but she gets pregnant. Now the clinician will not be able to continue the medication because the safety during pregnancy is unknown. So then the clinician will have 3 options: either continue with the drug and putting the fetus and the mother at risk. Number 2, withdraw the drug and put the mother at risk. Or number 3 is abort the baby. All of the 3 options are unwanted. Why such situation happens? Because we do not know which drug will cross the placental barrier, which is essentially a gatekeeper during pregnancy. So it's again a very peculiar organ which forms only during pregnancy, and it works as a protector for the growing fetus. A drug will known to be teratogenic only if it crosses the placental barrier and goes from the mother to the fetus to a certain enough concentration. Now, to understand that which drug will cross that placental barrier, but without involving a pregnant woman, we are trying to develop a platform that is known as Placenta-on-chip. We are working that in collaboration with Professor Deepak Modi from NIRRCH, ICMR, Mumbai, India, with his lab member Anshul Vire and my lab member Saurabh Mukherjee. So when we talk about the placenta-on-chip, first let us think of what is known as organ-on-chip. So an organ-on-chip is essentially, as you can see here, is a microfluidic structure made of generally some kind of polymer with this kind of micron scale, that is the size of your hair, of this micron scale kind of structure, and there you put the various different cell types. Essentially, we will call something as organ-on-chip if it can capture at least one structural aspect and at least one functional aspect. So in our placenta-on-chip, we will keep this criteria in mind. So let us see what structural aspect we are going to capture. For that, let us try to look at what is the structure of the placenta. So if you will look carefully, you can see that here the maternal artery is kind of releasing the blood in this huge pool here, where we have the fetal artery and fetal vein are here. Essentially, all the transport is taking place. So if I kind of zoom in this portion, it will look like this. So this yellow or this orangish portion is essentially the maternal blood, which is separated from the fetal blood, which is this fetal vein and artery, are by the endothelial cells, fetal endothelial cells. And then the maternal side, we have this trophoblast and the syncytiotrophoblasts. So essentially, maternal bloodstream and the fetal circulation, they are separated by the trophoblast and the syncytiotrophoblasts coming from the maternal side and the fetal endothelial cells. So that is exactly what we have tried to mimic. What we have here is a membrane mimicking the basal membrane. Then we have two chambers. One side we have the endothelial cells, another side we have the trophoblast cells. So it looks something like this. And the membrane is coated with the collagen for the cell adhesion. And then we add the relevant cell types from one side. Then when they are attached, we flip it. We add the other cell type from the other side and we allow them to grow, and we do all our experiments. So this can fit into one well of a 6-well plate, that is enabling us for doing a large scale or high throughput drug testing. Also, as you can notice here, that it is a flowless structure. So essentially it is out of the shelf, you can use it. So the users don't need any special training to use it. They can just follow the user manual and test it. Now, as I said that, okay, structurally, first I need to check structurally and functionally. So we ask the question that what happens in the real placenta, that the trophoblast in the presence of forskolin gets converted into syncytiotrophoblasts. Does that happen in our case? Syncytiotrophoblasts is known to produce human chorionic gonadotrophin, HCG. Is that happening in our case? And finally, does it show this selective permeability? So we will check for these three criteria. So as you can see here, we have the cytotrophoblast, a nice monolayer. We have a endothelial cells, nice monolayer, sandwiched two sides of a membrane, which you can see here, two sides of a membrane. And because these trophoblasts, they need to form the tight junction. So you can see that the tight junctions are formed here. So essentially they have created a nice continuous layer which will work as barrier to the transport. Next, as we said, that do the trophoblast get syncytialized? To do that, we add something known as forskolin. So when the forskolin is added, these trophoblast cells, which happens also in vivo during the actual pregnancy, these trophoblast cells, they are supposed to get fused so that you can see only the nucleus, but not those boundaries that you have seen you have seen previously. And when these trophoblast cells become syncytiotrophoblasts, they are supposed to secrete HCG, human chorionic gonadotropin, which you can see here in the graph, as well as you can see in this typical pregnancy strip that generally we use, which is nothing but to measure whether the HCG is there or not. And that also we have used. We have collected the sample, put that in our we collected the sample from our device. We put that in a commercially available pregnancy strip, and you can see the two lines here, ensuring that yes, just like a real pregnancy, here also it is secreting HCG. Now, once we have talked about that, okay, structural, functional, now we need to look at whether it is working as a selective permeable barrier. So when I say that the selective permeable barrier, what do I mean? So from the maternal side, something should get transported intact. They should pass through the placental barrier without any hindrance. Then there should be another set of molecules that should not get transported, that will come back. Third, there will be some molecules which should which we call it reverse transported, that is should come from the fetal side to the maternal side, and finally something should get partially consumed and transported. So these four the criteria we have put, and to check these four criteria, what we have here are a small molecule, small fluorescent molecule known as FITC, which should be transported intact. Then we have a large large molecular size fluorescent molecule FITC-dextran, as well as the immunoglobulin IgM. These two should not be transported from the mother side to the fetal side. Then we have urea, which is a secreted molecule of the fetus that should come from the fetal side to the mother side as a reverse transported, and then the glucose that should be partially consumed. So as I have already described, that we have two chambers here. One is maternal, one is fetal. For the direct transport, we add FITC or FITC-dextran or IgM in the maternal chamber, and we look for the direct transport. For the reverse transport, we add urea in the fetal chamber, and we look for the reverse transport. So what we see here, when I add FITC, it's a small molecule, it should get transported without any hindrance. That is exactly we see here. In case of a membrane without cell, the transport started as soon as we add the FITC, and it goes. When there is a co-culture, two cell layers are there, the transport, there is a lag time, but then the transport goes on without any hindrance. The concentration in the fetal chamber increases exponentially. However, when we put the FITC-dextran, which is a large molecule and which should not get transported, as you can see here, again, no cell, it gets transported without any problem. But as soon as you put the placental barrier, you can clearly see that there is no transport happening. That is, the concentration in the fetal side is not increasing with time, confirming that our placental barrier that we have made is actually working as a good barrier to the transport of the unwanted molecules. Same thing we can see here for the immunoglobulin, the immune molecules from the maternal side. And we look for the urea, which goes from the fetal side to the maternal side. And that is what also you can see that we add urea in the fetal side. And with 8 hours, fetal side concentration goes down, maternal side concentration goes up, indicating the urea is getting transported from the fetal side to the maternal side. Next, we looked for the glucose transport, which is a facilitated transport. Some portion should get consumed and some portion should get transported. In vivo, we see that the mother side, the physiological glucose concentration is 4.4 millimolar, and the fetal side glucose concentration is 3.6 millimolar. Ratio should be 0.81. In our case, when there is no cell, ratio is close to 1, both the sides get balanced. But in case of the co-culture, we see that the value is 0.86, which is very close to the fetus to mother ratio that is observed in case of the physiological condition. Finally, as our goal is for the drug testing, so we have chosen a drug valproic acid, which is an anti-epileptic drug and known to be a teratogen that is causing harm for the developing embryo. So we used this drug valproic acid, and as you can see that there the integrity of the barrier actually remains unchanged. With VA or with another drug we have used, I'm not going into that details, but we can see that the there is no difference, essentially telling us that the barrier remains undisturbed. And that is exactly we see when we give the FITC-dextran, whether there is VA valproic acid or not, there is no difference in the transport process. That is, the valproic acid is not making the barrier leaky, that the HDAC will go from the mother side to the fetal side. Then why is it harmful for the growing fetus? We checked for the glucose transport, and there we can see that the glucose transport get compromised when you add the valproic acid. So the time constant, that's the time required for the transport, increases in the presence of the valproic acid compared to the without the drug. So essentially it is showing that the valproic acid is changing something that is hampering the glucose transport. And what is that something? And when we looked at, we realized that the something known as the GLUT receptor, which essentially takes the glucose from one side and gives to the other side, the expression of the GLUT receptor gets compromised, which causes the glucose transport compromised in the presence of this valproic acid. And is our result clinically relevant? We look at the National Library of Medicine, and if you look at valproic acid might increase the chance of low birth weight and the report of temporary low blood sugar level in the newborn. So essentially what we have shown is that valproic acid in our device, it shows that it reduces the GLUT receptors, the receptors that are responsible for the glucose transport. As a result, in the presence of the valproic acid, glucose transport goes down, which may explain why the valproic acid in real pregnancy, in vivo physiological condition, results low birth weight and low blood sugar level in the newborn. So where are we? What I have shared with you is that yes, we have created a device. That device is structurally mimicking placenta. It mimics the placental function in terms of the differentiation of the trophoblast cells into syncytiotrophoblasts, secretion of the relevant molecules, as well as it works perfectly well the way we expect the placenta to behave as a placental barrier. So does that solve all the problem? Are there any challenges? Yes, there are. Key challenges are placenta changes structurally and functionally during pregnancy that we have not captured. That one may want to capture what dynamic changes happens in case of the placenta. We have used the cell lines. It will be good to use the relevant primary cells, which will make our device more relevant. We have shown only the static device, which is basically our idea. The strength here is that, yeah, this is easy to use. Anyone can use it without any prior training. It does not have pump, tubing, etc., all of the peripheral. So it is user friendly. However, it does not capture the flow profile that is happening inside the placenta, which we are right now working on. And finally, this device, the whole idea of this device is that as a predictive model to comment on the which drugs are safe during pregnancy. So essentially we require the regulatory as well as the ethical concern regulatory clearance so that such devices can be used to talk about, to comment on the which drugs or which medicines a ailing pregnant woman can take to reduce her discomfort. With this, I would like to thank you for all your attention. No work is possible without the grant. So here are all the various grants that has been enabled us to do this work, particularly imprint from DST. Majority of the work has been supported by the imprint DST. As I have already mentioned, the work is done in collaboration with my friend and collaborator, Professor Deepak Modi, his lab member Anshul Vire, and my lab member Saurav Mukherjee. Saurav was assisted with Ishira and Sandeep. Thank you very much for your attention.